Hello and welcome to this Bioprocess International Ask the Expert webcast. I'm your host, Leah Rosen, the online editor for Bioprocess International. Before we get started, just a couple of notes. This webcast is being recorded and will be made available for replay in the multimedia section of our website. We've muted the audio lines, but we welcome you to type in your questions for our speakers at any time in the question and answer window. Your questions in the question and answer window will be only visible to myself, our moderator, and our speakers. After the presentation, the moderator will be asking our speaker your questions from the question and answer window. So thank you again for joining us today. It is now my pleasure to introduce the moderator, Tung Luong from Asahi Kasei Bioprocess.
Greetings, everybody. Thanks for the introduction, Leah. My name's Tung Luong. I am the Global Marketing Manager for Asahi Kasei Bioprocess, and I'm excited to share some great information with all of you. So first, I'd like to start our webinar just to go a little bit over our company, Asahi Kasei Bioprocess, and the 4 business units that we consist of. So the first business unit is the one on the left there, our purification business unit, which consists of our industry-leading Planova virus filter product line and our BioOptimal microfilter line. Next is our fluid management, which manufactures large skids for large biopharma and created our groundbreaking Motive inline buffer formulation skids, among other key products. The next one there is our biosafety testing services. This business unit consists of VirusSure, a CRO which services in virus clearance and other services, and Bionique, a mycoplasma testing service. Finally, our latest acquisition and addition is our biologics development and manufacturing business unit. And that consists of Planova, a CDMO focused on developing and manufacturing your products from start to finish. So today, I'm really excited to introduce our S20N filter for you. We would like to introduce you first, our first speaker. Our new virus filter, S20N, recently launched as a next generation cellulose virus removal filter. This webinar commemorates the launch of S20N. We're excited to introduce these speakers. So our first speaker is Kazuya Kobayashi. Kazuya Kobayashi has worked with Planova virus removal filters for over 10 years and is an expert on virus clearance evaluation at Asahi Kasei Medical. He's a project controller planning and coordinating the progress of several projects, including the Planova S20N launch project, based on customer requests obtained through his accumulated technical support experience. So let's start with Kaz.
Let me thank everyone attending this webinar today. I'm Kaz Kobayashi. I'm a manager at Asahi Kasei Medical. I'm located in Tokyo, Japan, and in charge of technical support for Planova customers and project controller of Planova S20N. Today, I'm going to present about Planova S20N, which is launched in 2021 as our novel filter. This is my presentation contents. First, I'd like to present about the development concept of S20N, and then quick introduction how we developed it. After that, Planova S20N key feature will be shared. Before my introduction about the development concept, let me talk about the brief history of Planova filters. We launched Planova 35N as the first virus removal filter in the world in 1989. And then 50N, 20N, and BioX have been launched. In 2021, our novel Planova filter, Planova S20N, was launched. S20N is also originated cellulose hollow fiber-based filter as 35N, 50N, 20N. There is related to the background of the concept. Here is the concept of Planova S20N. One of our existing products, Planova 20N, is recognized as the golden standard virus removal filter in the industry. The plus point of 20N is robustness. Robustness means that under various solution conditions, the similar behavior for flux and throughput thanks to the hollow fiber material originated cellulose. And also very stable flux, high protein yield, and good virus removability are well-known features. But 20N requires gold particle test, the GPT, as postuse integrity test. That is because the physical weakness of the hollow fiber. GPT is more complex than leakage test, and also it takes time. So, to improve the customer satisfaction, the concept of S20N is defined as following. For wide window filterability, the material should be same as 20N regenerated cellulose, and parvovirus removal grade are required from the industry. And also higher pressure resistance to have high flux and to skip GPT. Here we say no GPT. About the no GPT, we already have the product BioEX. BioEX does not require GPT for postuse integrity test. That is because the much higher elastic limit of the hollow fiber than the maximum transmembrane pressure as described in this table. And we validated no change in pore size distribution affecting virus renewability occurs within the operational pH and pressure range. And also we confirm the correlation between the parvovirus renewability and the leakage test results. So for no GPT, high elastic limit is the very key point. But this is the most tough or difficult challenge for regenerated cellulose. To break the challenge, we utilized AI technologies. You know, we have more than 20 years data of correlation between spinning condition and hollow fiber characters of regenerated cellulose. By consolidating the data, AI predicts the manufacturing condition for S20N hollow fiber with high pressure resistance. Of course, AI predict has some error, so we need some in-house evaluation and adjustment. Since then, finally, the optimum spinning condition for S20N hollow fiber was determined. On this graph, S20N has the higher elastic limit than 20N. This is the development history of S20N. In following pages, I'd like to show you some data about Planova S20N features. They are categorized as below: improved performance, the improved operational efficiency, and traditional Planova qualities. First, I'm going to share about the improved filterability of S20N. Here is the filtration data of plasma IgG, which is conducted in duplicate runs. The solution condition is 1% polyclonal IgG in pH 4 and low conductivity solution. S20N has almost double flux than 20N with very stable flux. 20N also has the stable flux, but the filtration volume in 12 hours, S20N is more than twice of 20N. This is also polyclonal IgG filtration data, but this one is a high conductivity solution. It is including 100 millimolar sodium chloride. pH and the protein concentration is very similar to the last page's one. In this condition, S20N showed almost twice filterability than 20N, and both filters have very little flux decay. In this page, I'm going to show the filtration data with monoclonal antibodies. We evaluated two types of MAB. MAB A is a low hydrophobic one, and MAB B is highly hydrophobic one. In both cases, S20N showed more than twice filterability than 20N on this table. But for both cases, S20N and 20N showed very stable flux like this graph. As I presented, S20N has higher flux and very consistent filterability for polyclonal IgG and hydrophobic MABs with stable flux. In the following page, I'd like to show the robust virus renewability of S20N. S20N has high virus removability for various viruses at the table. Even though there is a 30 minutes process pause, no viruses are detected in the filter rate of S20N. Not only the parvovirus, MVM, the PPV, but also the other viruses, including HIV. And we evaluated the virus removability with longer process pause and lower filtration pressure condition. With these studies, we confirm the great virus removability of S20N. Even though the combination of long duration of process pause, 3 hours, and also the lower filtration pressure, S20N showed complete parvovirus clearance. I think you find the improved performance of S20N in the previous pages. So I'd like to show the operational efficiency of S20N because of no GPT. As BioEX, S20N has high elastic limit, and when confirmed, S20N has a resistance to pore-size distribution shift, affecting virus removability within the operational pH and pressure conditions. Planova S20N has great virus removability after the harsh treatment, which is a combination of low or high pH, high temperature, and higher pressure than maximum transmembrane pressure. And we find the correlation between the virus removability and leakage test result. The result is described as leak value, which is measured by our own integrity test equipment, PLT. Because of this data, S20N does not require GPT, unlike BioEX. Thanks to no GPT, Planova S20N contributes to shortening the manufacturing process time. GPT, which is required for traditional cellulose filter 15N, 20N, 35N, it will take sometimes 1 hour or more for preparation and execution and judgment. And not only the no GPT, S20N shipped as the sterilized and wetted as ready to use. You mean that it's like the other Planova filters. Because of the no GPT and ready to use, S20N is very good filter for the improved operational efficiency. In the next 3 pages, I'd like to brief introduction of traditional Planova qualities of S20N. As I showed in previous page, S20N has very consistent and reliable filterability as 20N. S20N can also be used for longer filtration time. It is possible for you to increase the filtration volume or shorten the time with the same volume. And robust virus removability even if the solution condition changes. With our internal study, pH and conductivity has no impact on the virus removability. It means it is possible for you to change the solution condition to maximize the filterability without impacting virus LRV. And we confirm the great scalability and lot-to-lot consistency of S20N as this graph. Today, I presented the Planova S20N features described here in my session. Here is a summary of today's my presentation. Our novel Planova filter, Planova S20N, was launched, and I presented the key feature of S20N. Thanks to the high pressure resistance, the S20N has high flux and does not require GPT with these traditional Planova qualities. I have limited time today, so if you have any question, please let us know. We are happy to introduce S20N and conduct filtration study on your site with updated data of S20N. Thanks for your attention.
Thank you, Kaz, for your great presentation. We'll take questions at the end of this presentation coming up. Kaz cannot join our Q&A today, but I will field any questions directly about our filters. I'd like to introduce our next speaker, Jessica Waller. Jessica received her PhD in physiology and neurobiology from the University of Connecticut and a BS in biology from Stockton University. She has over 7 years with Celgene, a wholly owned subsidiary of Bristol Myers Squibb. She continues to play key roles supporting early and late stage portfolio products, serving as a downstream purification lead for bispecific assets and managing cross-functional collaborations. Welcome, Jessica.
Thank you for the introduction. Hello, everyone, and thank you for joining the webinar today. And thank you to Asahi Kasei and BPI for the invitation or the opportunity to speak today. So the focus of my presentation will be on Planova and flat sheet nanofilter performance, incorporating the new generation S20N for a molecule with a trispecific format. These studies were performed in collaboration with Asahi and include initial filter comparison studies by performing capacity assessments, followed by evaluation of MVM clearance across these different filters. So first, a brief background on multispecific antibody properties and challenges they impose for downstream processing and viral filtration. So although bispecific antibodies and trispecific antibodies have enhanced functionality with the ability for dual targeting and increased tumor selectivity, there are unique challenges associated with purification of these molecules in contrast to monoclonal antibodies. In addition to high molecular weight aggregates, many combinations of other product-related variants, including mispaired species, not typically seen in monoclonal antibody product pools, can form even with recently improved engineering approaches. So so these include free heavy chains, free light chains, homodimers, 3/4 antibody species or incomplete species, half antibodies, and other mispaired light chain, heavy chain species. A summary of these different types of mispaired variants are shown in schematics on the left and the table on the right, as well as the presence of process-related impurities such as host cell protein or HCP, residual DNA, and residual protein A that must be monitored and cleared throughout the downstream process. Viral clearance capability is another critical factor when developing a process. CHO cell-derived products have an inherent risk for viral contamination that can be introduced during production or through raw materials used, and thus can pose a risk of endogenous retroviral or adventitious agent contamination. And according to regulatory guidelines, the purification process should demonstrate robust viral removal capabilities of approximately 12 to 18 logs clearance of endogenous retroviruses and approximately six logs for adventitious viruses. And robust clearance of a unit operation is generally defined as greater than 4 logs. So considering these challenges encountered with multispecific antibodies and the unique panel of impurities associated with these molecules, we employ a 3-resin-based step approach in which the combination and types of polishing column resins can vary and are dependent on the molecule properties as well as beginning impurity levels. This is a schematic of the general bispecific purification process incorporating filtration and chromatography unit operations. So depth filtration is employed for clarification of bioreactor material. This is followed by protein A chromatography as the capture step from clarified cell culture material. This is followed by a low pH viral inactivation leading into the intermediate polishing step, and then the second or final polishing step. And the protein A and polishing steps do achieve some clearance of or removal of product and process-related impurities, as well as some orthogonal virus clearance. And following the second or final polishing step, the virus filtration through a nanofilter is performed, another dedicated viral reduction step in addition to low pH viral inactivation. However, the feed stream leading into this step can be characterized by high concentration and high conductivity, since the final polishing step is generally operated in bind-in-a-loop mode. In addition, residual product-related or mispaired variants, typically low molecular weight species, not removed by the final polishing step, can also be present in this feed stream. So all these characteristics could promote fouling across the nanofiltration step and potentially impact viral clearance. So thus there is a need for a robust viral filter with high compatibility as well as high capacity and scalability for these more complex configurations. So an evolution of Planova filters and their characteristics are shown on the bottom with BioEX and S20N and the new generation S20N able to operate at higher pressures and possess the added advantage of air diffusion integrity testing as opposed to gold particle testing required for 20N. So we assessed performance of Planova 20N, S20N, BioEX, and a flat sheet nanofilter for comparison for purposes of multi-specific antibody toolbox capability for viral filtration, as well as viral clearance capability for an IgG molecule with a tri-specific format. So the approach we took was an initial assessment of capacity, comparing Planova 20N, the new generation S20N, BioEX, and a flat sheet nanofilter configuration. Next, clearance studies using these different filters were performed with MVM used as the model virus, typically used during viral clearance studies. The schematic of the general setup is shown on the left, indicating the flow path and valve locations from the reservoir to the viral filter, and data was collected with Asahi acquisition software. So for initial capacity assessments and clearance studies, control material or representative polishing column 2 eluate material with a concentration of approximately 6 mg per mil, pH 7.2, and conductivity of approximately 30 millisiemens per centimeter was used. And 20N was operated at 12 psi, S20N at 28 psi, and both BioEX and the flat sheet nanofilter at 35 psi with target throughputs of 800 liter per meter squared for the Planova filters and 1,000 liter per meter squared for the flat sheet nanofilter. And for these studies, the feed material was sterile filtered prior to nanofiltration, and all Planova filtrations were performed in the absence of an inline pre-filter, whereas an inline pre-filter was incorporated for the flat sheet filter. So again, for these initial filter comparison studies for this tri-specific antibody under representative polishing column 2 eluate conditions and using Planova 20N, S20N, BioEX, and the flat sheet nanofilter, illustrated here is a flux summary graph and table. So all filtrations were able to achieve the target volume throughputs and demonstrated high capacity, with S20N exhibiting the highest flux of 107 LMH among the Planova filters, and 20N about half as expected due to the lower operating pressure, together with very minimal flux decay. Vmax estimations reveal that S20N can reach a very high capacity, and in addition, BioEX did demonstrate consistent performance for this class of molecules as seen in previous studies. In addition, all 3 Planova filters did demonstrate linear throughput over time, which is an indicator of steady flux. And finally, filtration performance was greatly improved for the flat sheet nanofilter in the presence of an inline pre-filter due to the higher conductivity of this feed stream. So without an inline filter, an almost immediate and significant flux decay was observed. So these studies did confirm robust performance of the Planova filters, notably S20N, in the absence of an inline pre-filter. So based on the capacity assessment, we next targeted 600 liter per meter squared volume throughput, or approximately 3,500 gram per meter squared mass throughput for MVM clearance studies using these 4 filters for this tri-specific molecule. Above is the experimental scheme in which 3 separate equal filtrate fractions were collected and a 60-minute pause implemented prior to a buffer flush of approximately 1 holdup volume for the Planova filters and approximately 50 liter per meter squared for the flat sheet nanofilter. All fractions, including the buffer flush as well as the pooled filtrate of all fractions, were assessed separately for MVM clearance by the TCID50 assay. Also, preliminary qualification, including cytotoxicity, interference, and virus recovery experiments, confirmed the lowest dilutions that could be used for assay of the filtrate samples with no matrix or protein interference. So illustrated here is a summary of the flux performance across the 3 Planova filters in duplicate for this tri-specific, with operating pressures of 12 psi for 20N, 28 psi for S20N, and 35 psi for both BioEX and the flat sheet nanofilter, the same operating pressures as in the previous study. So all filtrations did reach the target volume throughput of approximately 600 liter per meter squared. However, BioEX and the flat sheet nanofilter exhibited the highest flux decay, up to 67% for BioEX and 44% with the flat sheet filter, even in the presence of an inline filter for this flat sheet configuration. There was consistent flux performance across 20N and S20N, although slightly elevated decay for S20N in the absence of an inline pre-filter. And for MVM clearance studies, the load material had MVM concentrations ranging from approximately 5.6 to 5.8 log 10 TCID50 per mil, or an approximately 8 log per filter MVM challenge target. And the TCID50 assay was performed for each filtration sample. So despite the 60-minute process pause prior to buffer flush and the high loading, all filters did achieve pool LRVs of 4.4 to 5.5, highlighted in the right column of the table, and thus robust MVM removal. So to summarize, with the challenges that exist for these more complex molecular configurations and the additional panel of impurities that can arise and must be monitored throughout the downstream process, nanofiltration performance and thus viral clearance can be impacted with these more complex feed streams. So we demonstrate robust S20N performance as well as MVM clearance capability for a molecule with a more complex format with a high conductivity feed stream and some residual product-related impurities leading into the nanofiltration step. So first, a filter comparison study was performed for this tri-specific antibody using Planova 20N, S20N, BioEX, as well as the flat sheet nanofilter in parallel with a pre-filter inline just for this filter. S20N did exhibit the highest capacity with the least flux decay, whereas the flat sheet nanofilter required an inline pre-filter to mitigate almost immediate and significant flux decay and reach the target volume throughput. In addition, all filters were able to achieve or attain robust MVM clearance. So these studies suggest that hollow fiber nanofilters and the new generation S20N do demonstrate robust performance and clearance capabilities for more complex molecular configurations, multi-specific antibodies in this case, and with 20N and BioEX also possessing the added manufacturability advantages in terms of processing time and integrity testing. So I'd like to thank the Downstream Process Development Group at BMS for their support of this work and our great collaborators at Asahi Val, Eric, Zoe, Christine, and Daniel for their support, their assistance in study design, and for generating all the capacity and clearance data. And thank you for your attention.
All right, thank you, Jessica. We're going to begin our Q&A. So if you have any questions, please type them into the question area, and we'll see if we can get to them. In addition, we have some contact information here. So if you have any additional questions for Kaz, or you need support, evaluation support, a meeting, we're happy to meet, and you can contact us there. So that AKB can ensure that we get to you quickly. So with that, let's move to the questions and answers. I'm going to answer some of the questions here that are kind of specific to Asahi and S20N. So I see one that's kind of just asking about the AI technology that we used, and that's an in-house, so I could say, AI technology. So it's not something that you can get off the shelf, unfortunately. Next, I see another one about how do you sterilize the filter? The filter already comes pre-sterilized, so it just comes ready to use. So when Kaz said it's ready to use, you just open it. It already comes pre-wetted. You can hook it directly up, and it works right away. And then I think that is most of the questions that are specific to S20N. We have a couple questions here for Jessica. Let me pull up one. So what are the dips of flux observed in BioEX? I think it was maybe your 5th slide or something that had a couple of just small drops with BioEX.
Sure. So I think they must be referring to the initial study. That's probably just related to pauses during the process. It's not specifically related to actual performance.
Okay. Yeah, I think it looked like maybe just small little pressure variations or
right.
extra drops coming through.
But nothing significant.
Yeah, I see here's another question. Were the loads on the viral filter stored frozen prior to processing?
Yes, so they were subjected to one freeze-thaw cycle, and we didn't see any issue with performance due to that freeze-thaw cycle.
Okay. Here's a question. What would be a driver for you to choose S20N over BioEX, since maybe they look a little similar?
Sure, that's a good question. Again, we'd have to gather more data maybe for this specific class of molecules, but again, it would probably be related to performance, minimal flux decay. We did see better performance in terms of the decay for S20N. So again, we'd have to confirm this with other classes of molecules as well.
And I guess speaking of that flux decay, I guess there's a question on the general range of the high molecular weight and HCP in your feed stream. Do you think that may have interfered with with that to create that flux decay?
In this case, I don't believe it would be attributed to that. This was a fairly pure product pool leading into this step. So high molecular weight was below 1% and HCP well below acceptable levels. It was well below 10 ppm. So in this case, I don't believe that flux could be attributed to any impurities. We didn't see any other aggregation issues going into this step. So it could be potentially the higher decay we saw in the spiking studies could be due to that virus spike. But again, we were targeting much higher than we would normally do. So we went up to 600 liter per meter squared. We would normally target about a 300 liter per meter squared volume throughput, in which at that point, the decay wasn't very severe.
Okay. And then I see something here for what is the spike dilution used for the MVM study?
Sure, you mean in terms of, if it's in terms of volume spike dilution of 0.05%?
Okay. Someone's asking you to turn on your camera.
Oh, sure.
maybe people can see you a little
Sure.
bit better.
Sorry about that.
No problem. Let's see. Was the polishing step prior to the VF operated in binaural loop?
Yes, it was.
Okay, I do see a couple specific ones about the filter itself. One, the sterilization method here, there's another question on the sterilization method that is just autoclaved. So in this particular filter, it is autoclaved. There's another question here, what component makes S20N superior with higher elastic limit? So it's just a new manufacturing method. So the material is still regenerated cellulose, but with a new manufacturing method that we've come up with, it creates for a stronger elastic limit. So that allows for higher pressures and allows us to remove that gold particle test that we wanted to remove. There's a question here for you, Jessica. How long can S20N be paused without impacting throughput? I mean, I think you did a short pause and a really long pause.
We actually only did one pause. We only tested a 60-minute process pause prior to buffer flush.
Okay.
So we didn't do any longer duration pause studies. We'd have to investigate that further.
Yeah, I think internally we've done up to 3-hour pause for S20N and still have not seen an impact. So that's usually the maximum amount that we kind of study for. I mean, I guess you could go longer, but I think if there usually is breakthrough, it actually happens earlier in that pause, right in the beginning, as opposed to duration. It kind of just stops flowing after that. Here's another question. Does the load concentration impact the flux or not?
So for these particular studies, we only used our control material was approximately 6 mg per mil. We didn't test various load concentrations for this trispecific molecule, but we have performed previous studies with a bispecific molecule in which we did vary load concentration. And we went up to 30 mg per mil load concentration and didn't see an effect on flux performance.
Great, thanks. There's another Planova specific one, which is for S20N. They appreciated the comparison between 20N and BioEX. Is S20N intended to be an improved version of 20N or BioEX? And it is intended to be the next generation of 20N. So we hope that people will consider S20N in place of 20N. Of course, 20N has its place if you're still using it in your processes. And for BioEX will have its place in our portfolio as a kind of working for niche products or high concentration products. That's where we kind of see that filter sit in line. Let's see. The questions are coming so ridiculously fast. It's hard to keep up here. So I'm sorry if we're not going to be able to answer every single one of your questions, but we'll try to get to a few more here. So this says, Jessica, the breakthrough that you experienced on 20N, where was this breakthrough detected? Post-buffer chase, only in a pooled sample?
So, yeah, that was a slight impact again from that process pause, the 20N. We're still awaiting data on the actual LRV value for that buffer flush sample, but yeah, compared to the other filters, I think 20N is more sensitive to process pause in terms of breakthrough.
Right. So yeah, I think there's another question that was kind of, do you think that's attributed to pause or filtration or chase? But it sounds like we don't have a complete answer on that just yet. This is a silly, interesting question. Why do we call it S20N? It's a little confusing to 20N. Yeah, we wanted some relation to 20N. There is, 20N is one of the most popular, most used filters out there in the market. It's basically been the #1 filter out there for a really long time. And the S really stands for superior, simplified, and secure. So we wanted, some relation there. but letting people know that it is related to 20N in some way and what people use. This is another question. Since S20N no longer needs GPT, is Triton X100 no longer needed for post-use integrity test? That is correct. You do not need to use, you don't need to do a post-wash any longer. You can just do your buffer chase and then you're done. So this really simplifies the process afterwards. Yeah, I think that might, let's see, maybe we can handle, I think that might cover most of the questions. If we didn't get to any of these other questions, we can, we'll email them to Jessica, and our Acai team will also reach out to all of you who've answered questions just to make sure that we answered them correctly. So with that, I think, thank you everybody for attending. Thank you so much, Jessica, for taking the time to sit down with us and reach out to all our interested customers out there. And again, please click on the email, click on the LinkedIn, click on the website. Anything for Jessica, you can send that email to that TCS support, and we'll forward that to Jessica so that she can respond to you. Okay, thank you so much.
Thank you, everyone.
Okay.